Welcome back to Virology, and today we are going to talk about how viruses with DNA genomes replicate those genomes. Remember, the genome is in the particle. So today we're going to talk about these viruses, class 1 double-stranded DNA, class 2 single-stranded DNA, and class 7 double-stranded gapped DNA. And we're going to talk about how those are replicated. Now, obviously, you have to replicate the DNA to make more particles, right? That's very clear. Not a difficult concept. But what I want you to appreciate is, and this comes from the last session where we talked about transcription, is that DNA replication is always delayed after infection. It cannot start immediately. A few things can start immediately after release of the genome into the cell, for example, but not DNA replication. Why? Because you need at least one protein and sometimes more to make DNA, and we'll talk about that today. So viral DNA replication always delayed, and that's why those viruses, SV40 and adenovirus and herpes virus, they have transcriptional programs to delay the production of capsid proteins until DNA synthesis begins. Because if there's no DNA and you make capsid proteins, you're going to make empty capsids and they're going to be non-infectious. So just as for RNA, there are universal rules for DNA replication. And here we have on the upper left is a template, 5 to 3 prime, and there's a primer going from 5 to 3 prime, hybridizing to the template. And of course, the bases are added at the 3 prime end. So DNA is synthesized by template-directed incorporation of DNMPs. So DN could be A, C, G, T for DNA, monophosphates into the 3 prime end of the DNA. And the DNA is always synthesized in a 5 to 3 prime direction. It is what we call semi-conservative replication, which means there are 2 daughter strands. We'll look at that in a moment. I don't know why they're called daughter and not sons. I didn't name it. It was a long time ago. I don't even know why they call them daughter. They should be progeny or complement or whatever, but there you go. So don't complain to me. I didn't do it. And replication of DNA begins at specific sites that are called origins. We'll talk about origins or origins of replication, or ORI. That's where the DNA synthesis begins. Of course, the DNA is made by DNA-dependent DNA polymerase, DP, plus other proteins are always needed. And it can be primer-dependent or primer-independent. So either way works. So I always used to teach in this course that DNA replication is primer-dependent. But a few years ago, a paper was published showing that there is at least one DNA polymerase that's primer independent. This is the experiment that was published that shows that. This is a time course. You see on the x-axis is time, and the y-axis is incorporation of bases, monophosphates into DNA. So that means DNA synthesis. And here we have 2 different polymerases. 2 different DNA-dependent DNA polymerases. One is T7 DNA polymerase, T7 DNAP. T7 is a bacteriophage. In fact, the other is a bacteriophage as well. And you see, without a primer, this polymerase can make no DNA. And once you add a primer, T7 DNAP plus primer, you get the red curve, primer-dependent polymerase. This is what most polymerases were observed to be over the years. But a phage called NRS1 was identified a number of years ago. This phage infects bacteria that live at the bottom of the ocean near those vents. You've seen these vents that all the stuff from the interior of the earth is coming out because there's hot water in there spewing out, and there's all kinds of life around the vents. Amazing, weird life. Remember, huge pressures, no light down there, and there are microbes. There's microbial life. There's all kinds of weird crabs and worms. And of course, there are viruses that infect these bacteria. And NRS1 is a phage that infects bacteria around these deep sea vents. We know this because people go down there and look at it in those little submarines that, if you're claustrophobic, forget it. You're never going to survive there. But this polymerase, NRS1 DNAP, without a primer, that's the blue curve, it works perfectly well. And if you add a primer, it doesn't really make a difference. So it's a primer-independent polymerase. So DNA polymerases don't have to be primer-dependent. DNA polymerases look very much like all the other three kinds of polymerase. So we've talked about RNA-dependent RNA polymerases. Here it is in red. I'm sorry, on the bottom in blue, that's the poliovirus RNA-dependent RNA polymerase. And remember, it looks like a right hand with the active region in the palm, and the fingers and thumb. For polio, they touch and they encircle the active site. You can see the fingers and thumb domains touching. There's the active site with the yellow beta strands. And here on the top is a DNA polymerase, DNA-dependent DNA polymerase. You can see the thumb and fingers are not touching. It doesn't matter to you. It's just something that is. And here's the active site in the yellow beta strands. And the double-stranded DNA template goes in, it's melted, and then one strand is copied, and then the other is copied as well. And the same kind of catalysis happens for DNA as for RNA. We saw this for RNA, the two-metal mechanism of catalysis, where two metals are coordinated by two aspartate residues in the active site. The two magnesiums they anchor the triphosphate and allow the series of attacks that allow release of two phosphates and the formation of a phosphodiester bond between the new base and the previous one. So that's a phosphodiester bond there between the C and the A, and a new one is going to form between the A and the T. So two-metal mechanism of catalysis. The cell also provides help. It's not all viral. And as I said before, DNA synthesis always needs at least one protein, maybe more. We'll see examples of that today. And that's why DNA synthesis is delayed, because it takes time to make those proteins. Viruses with small genomes require more host proteins. Why? Because they can't code a lot of proteins in their own genome. They're too small. You're limited in coding capacity. So more host proteins are needed. And as the genome of the virus gets bigger, you need fewer and fewer host proteins. And then there are many more viral proteins that participate in DNA synthesis. And in fact, the herpes viruses and the pox viruses encode all the proteins they need for DNA synthesis and for the production of dNTP substrates. So remember, when you make DNA, you need to use dNTPs to incorporate into the polymerase. These are the dNTPs here. And those have to be made in the cell. Our cells make those. We have all the enzymes to do that. And some small viruses simply cannot encode those enzymes, but herpes and pox viruses do. They can make their own dNTPs. The polymerase itself sometimes comes from the host. For viruses with small genomes, they do not encode a DNA polymerase. The genome is not big enough, and we'll see an example of that today. But they do encode proteins, it says here, that orchestrate the host. That means they direct the host to copy their genomes, because it's hard to be recognized if a small DNA genome goes into a big cell nucleus with lots of nuclear DNA. How do you get the attention of the DNA replication system of the cell? So that's what orchestrate means. And these include papillomaviruses, polyomaviruses, and parvoviruses. We'll talk about those today. But the large DNA viruses encode much of their own replication system. Adenoviruses encode a DNA polymerase, but not everything, whereas herpes and pox encode everything needed for DNA synthesis, including a DNA polymerase. So that's where the DNA polymerase comes from. And there are other proteins involved in DNA replication. There's the DNA polymerases, of course, the DDRP. There are accessory proteins. It could be viral or host. There's typically an origin binding protein. The origin of DNA synthesis is where synthesis begins, and there are proteins that bind to it. You'll see in a moment. There are helicases. They unwind the DNA. There are exonucleases. If the polymerase makes a mistake, and all polymerases do, the exonuclease will cut out the wrong base and it can be fixed. And then, of course, the enzymes of nucleic acid metabolism that make the monophosphate, precursors like thymidine kinase, ribonucleotide reductase, DUTPase, etc. Those are all necessary for DNA replication. So a rather complicated process. Our first question is, which statement about viral DNA synthesis is not correct? Large DNA viruses encode many proteins involved in DNA synthesis. Small DNA viruses encode at least one protein involved in DNA synthesis. Viral DNA replication is usually delayed after infection because it requires the synthesis of at least one viral protein. All DNA polymerases are primer dependent. Well, I thought this would be 100% actually, but there you go, 89%, not bad. All DNA polymerases are primer dependent. That's not right, right? I showed you, spent a couple of minutes on a slide showing you primer independent polymerase. Everything else is right. Well, large DNA viruses encode many proteins involved in DNA synthesis. Absolutely, it's on one of the slides. Small DNA viruses encode at least one protein. And viral DNA replication is always delayed. I guess the word always made you say that was wrong, right? And I tell you to be careful of always. Yeah. I mean, I'll take always out, because it's delayed. It would be correct. Maybe that would get rid of that 9%, right? I've been teaching this for 15 years, and I discover stuff every year. So the genomes that we're going to look at today, they're different structurally. And so that presents a challenge. How do we copy them if they're so different? So we're going to talk about single-stranded DNA viruses. So an example is adenovirus-associated virus type 2, a parvovirus. This is an interesting virus because in the last lecture of this course, we're going to talk about using viruses to cure diseases, right? Gene therapy. And parvoviruses play a big role in that. They're single-stranded DNA viruses. Then we're going to have double-stranded circular DNA viruses, SV40, adenovirus, linear double-stranded DNA, herpes viruses, linear double-stranded DNA, but much bigger. So adeno, 35,000 base pairs, herpes, 150,000. Then we have the pox viruses, 200,000 base pairs. And you see on this picture, we have origin binding proteins. These are viral proteins that bind the origin. They're in red. And then we have cellular replication proteins and viral replication proteins where they interact here. But we'll go into these in some detail. There are two general mechanisms of DNA synthesis. There is synthesis where you have a replication fork, and there is synthesis where you have strand displacement. So replication fork is basically just what it sounds like. The DNA is replicated on both strands, and as a consequence, the DNA is separated and looks like a fork. So we have synthesis going on in both strands, both the plus and the minus strand. And so here you can see we have primers being made in green. These are RNA primers that are made first, and they prime DNA synthesis, which is shown in red and orange. And that can happen on both strands in a 5' to 3' direction. And that happens with these viruses here, papilloma, polyoma, herpes, and retroviral proviruses. So retroviral provirus is the viral DNA integrated into the host. So our host cell does this kind of replication fork. And then on the right, adenoviruses and parvoviruses do strand displacement, where one strand is copied first. So we have a primer-dependent copying of the negative strand, and that new copy displaces the positive strand. That's why we call it strand displacement. And then that top strand will eventually come off, and that will be copied on its own. So both strands are copied. That's what we mean by two daughter strands. Both strands are copied. That kind of replication requires a primer. It can be a protein for adenoviruses, or it can be a DNA as for parvoviruses, and we'll see examples of that today. Now, when you have DNA replication, in many cases you have what we call a 5' end problem, right? And that's shown here. So on the left we have a double-stranded linear DNA. We have, both strands are going to be copied eventually to give 2 daughter strands. And so the original strands are dark blue and light blue. So each of those are separately copied, and you end up with a complementary strand to both. And their RNA primers are shown still there because they remain there for a bit. But the RNA primers are always removed because you don't want RNA-DNA hybrids. You want all DNA. So the RNA primers are removed. And the problem is, the end problem is, when you're at the end of the DNA, you can't fill in the gap made by removing the RNA, right? Because you can't put another primer there because it would just put RNA back and then you'd remove it and you'd be in a circle. So that's the 5' end problem. And there are several ways that you can get around that. And when we go through each of these replication stories, I'll point that out. So let's start with SV40. SV40 has a circular double-stranded DNA genome about 5,000 bases in length. So it's pretty small. 5,000 bases, pretty small. It has a single origin of replication. It's called ORI. And DNA synthesis occurs via a replication fork in both directions from the ORI. So we call it a bidirectional replication fork. And what happens is replication begins at the ORI, and you have denaturation of the double-stranded DNA, and then synthesis of the new strands, which are shown in red here. And that fork grows bigger and bigger until finally you have 2 complete double-stranded DNAs. And you'll see each strand is copied. The plus strand and the minus strand are each copied separately. So you'll end up with 2 copies where you had 1. And on the top is a series of electron micrographs, which were the initial proof that there is bidirectional replication from an ORI. What they did is they took SV40 DNA and they cut it with a restriction enzyme that would cut near the origin once. All right? And so now you linearize the DNA, and then you incubate it with whatever you need to replicate it, DNA synthesis extract, and you watch the bubble grow. And you can see the bubble is starting in panel A right there. So that's the origin of replication, and then it gets bigger and bigger and bigger, and it's getting bigger in both directions. You can tell that with reference to the end of the DNA. So bidirectional replication by a replication fork. So here's a bigger picture of what's happening with the bidirectional replication fork and why there's no end problem in this case. So here's the top strand, and here's the bottom strand, or the plus, or the minus, right? Because the plus is the same polarity as mRNA. So they're 2 different colors. And there's the origin of replication. And remember, the DNA was denatured initially at the origin, then the denatured bubble gets bigger and bigger. And so what we have here are 2 leading and 2 lagging strands. Why do we call them that? Well, the leading strand is named that because the polymerase can make an RNA primer right there at the origin and synthesize continually in a 5' to 3' direction because the template is 3' to 5'. So that's the leading strand. It's continuous. There's another leading strand on the bottom strand as well, right here. But to the right of the origin, you have the lagging strands because there the synthesis has to happen in a 5' to 3' direction, but the template is not exposed until the fork gets bigger. And so these are not continuous syntheses. And so this little one was made when the fork was small. And then as the fork got bigger, the next one was made, and the next one, the same thing happens on the other strand. And that's because to the right of the ORI, you can't do continuous DNA synthesis. And eventually those RNAs are removed, and then you can fill in the gaps with the DNA polymerase, so there's no end problem. Because there is no end on a circular DNA. That's the solution to having an end problem, is not to have any ends. And our DNAs are not circular, so we have end problems that are solved by telomerases, which we'll talk about later. So semi-discontinuous DNA synthesis because there's a leading and lagging strand. The leading is continuous, the lagging is discontinuous, and it's a bidirectional origin. No end problem. This is what the origin of SV40 looks like, just to give you an idea. So the origin is right there at base 1, which is right next to base 5,243 in the genome. So that is where DNA synthesis begins. The origin is right next to some transcriptional control regions. There's the early promoter, which you may remember is on early in infection, and this is an enhancer for the early promoter. And this below here, expanded, is the core origin. And you can see it has repeats, 27 base pair, perfect repeats. It has AT-rich elements, and so forth. Now we'll talk about what these are doing in a moment. Here's an overview of what happens initially. So we have a double-stranded DNA at the top. There's the origin of replication. And the origin specifically binds an early viral protein called large T, LT, or just T antigen. There are actually a couple of different kinds of T, so we say LT. But if I say T antigen, this is what I mean. And 6 molecules of T antigen bind on either side of the origin. There are binding sites in the DNA for that protein. That protein denatures the DNA. It separates the strands to get DNA synthesis going. And so that is an ATP-dependent process. You can see there are lots of ATPs bound to large T. And then to get denaturation of the DNA, that needs energy. And that's hydrolyzed. ATP hydrolysis provides that energy. Now you have a denatured part of the double-stranded DNA right there. Now, large T also binds a cell protein called RPA, replication protein A, which binds to the single strands and keeps them from reannealing. Because if you had a single-stranded region on both sides, it would just rebase pair if you didn't prevent it. So those proteins prevent it. So T brings in that protein, it recruits it essentially, and T denatures the DNA. So we say that T has 3 to 5 prime helicase activity. Remember, helicase is an enzyme that can unwind double-stranded DNA. So that's the first step. So T antigen is key in getting this going. T antigen also recruits polymerases from the host cell. There are a couple of different DNA polymerases that participate in this process. And so, for example, there's one called Pol alpha primase, which makes the RNA primers, and it also can make short DNA fragments. That binds to both RPA and LT. So LT is recruiting the cellular machinery. That's what I mean by it orchestrates DNA synthesis. These proteins are binding to LT. So now we have a polymerase alpha in there, and you can start to make short RNA primers. Those are shown in red there on both strands. That can happen. And then you recruit in a few other cell proteins, and then finally polymerase epsilon, which will make very long DNA. The polymerase alpha primase can only make short DNAs. The longer polymerase comes in and makes very long DNAs to complete the copying of the viral genome. And this is a topologically complex process happening here. Here you can see a molecule of Pol A primase on one strand, which is going to synthesize little RNA primers, and then it's also going to be on the other strand as well. And these double strands will get bigger and bigger. The bubble gets bigger and bigger until it covers the entire genome. And you remember, you're going to have leading strands and lagging strands going in the other direction. You're going to have RNA primers initially present, and those RNA primers are going to be removed. The RNA primers are shown in green, which may be hard to see here. But the RNA primers are removed by an enzyme called RNase-H. The gaps are filled in, and then the DNA is ligated. So you have a completely double-stranded molecule with no gaps in it. And that's what you get at the end. And this is a picture of what's happening here at one of these replication forks. Here's large T, which is going to move down the DNA and denature it. continuously. We have one strand bound to RPA to keep it single-stranded. And you can see DNA synthesis is occurring there. The red is the other strand. The green here and here is the RNA primer. And the same thing is going to be happening on the other strand as well. So amazing that this actually works. There are cool movies of that, by the way, that you can find that illustrate that completely. Now, when we're at the end, we have two double-stranded DNAs. There's another problem, and that is to get them separated. So first of all, when this DNA, covalently closed circular DNA, is highly twisted up, and as it's copied, it gets more and more twisted. So as you denature the DNA there at the origin, the unwinding causes the rest of the DNA to twist around itself. And eventually that would stop DNA synthesis if it weren't relieved. And so there are enzymes called topoisomerases that will cut one of the DNA strands, and kind of like cutting a twisted up elastic band. It all unwinds, and we now have what we call relaxed supercoils. So you go from this overwound situation to one that's relaxed. The DNA is cut and then it's religated together because you don't want to leave that cut in there. And that can happen multiple times during DNA synthesis as needed. But at the end, you now have a very complicated molecule, which is basically two copies of the original DNA, sorry, 4 strands, but they're still intertwined. They're like 2 circles joined. You cannot separate them at the end. You don't really see it in that drawing, but the 2 circles of double-stranded DNA are like 2 rings caught in each other. So what do we do? Well, topoisomerase 2 cuts both strands so that the DNAs can separate. So it will cut both strands of this 2 DNAs, and now you have 2 double-stranded DNA templates. And again, both strands have been copied. So this is often called resolution. At the very end, you separate the products away from each other. And now you have 2 genomes where you once had 1. All right, what did we do? All right, all of the above, they're all right. Almost 100. T antigen binds and unwinds the ORI. That's correct. Replication is bidirectional from a single ORI. That's correct. But people who pick that, other things are correct also. 5' end. So problem is solved because there's a circle. There is no end. The leading and lagging strand synthesis, they're all right. All right, let's do now parvoviruses. These are single-stranded DNA genomes, and that's what they look like. They're mostly single-stranded, and each end has a funny T structure. It's called a terminal repeat because it's the same sequence at both ends. And at the bottom is what it looks like. It's actually a part of the DNA that can base pair with itself because the genome is single-stranded, it can do that. And you see here's the 3' end, and all this part is base paired, and then the very middle is base paired as well. So that's where you get this T structure from. And this 3' end is, yes? Does the little end have to be mostly GC have anything to do with stabilizing the bonds? Yes. Yeah. So GCs, as you know, are more stable than ATs, and whenever you have a lot of GCs, that means it's going to be stable. If you remember from the origin sequence I showed you, there's a lot of ATs, which makes it easier to denature because that's what you need to do, right? So this hydroxyl is going to be the primer for DNA synthesis. So this is a primer-dependent reaction. This is going to be done by the host cell. Our DNA polymerases require primers. So any viral genome that utilizes the host cell polymerase is going to be primer-dependent as well. So let's see how this works. So on the left is the schematic for what's going on. So there's our genome on the second line with the 2 T ends. They are base paired, and above it is what it would look like if you denatured each end, right? If you melted the structure. And I do that to show you the labeling. They're both inverted terminal repeats, and this shows you what that means. So here we have A, B, C, A prime, D. So the A and A prime are complementary to each other. They will base pair. So anything with a prime will base pair with its complement. Here at the other end, we have A, B, C, A, D, which is the same sequence, except inverted. So it's a repeat of the same sequence, and it's inverted. Inverted complementary repeat, A, B, C, A prime, B prime, C prime, A, D prime. All right, so that's how you get the base pair forming. So you see A base pairs with A prime and so forth. This molecule gets into the cell nucleus, and the cell sees it as single-stranded and wants to repair it. Because single-stranded DNA is not appreciated in the cell nucleus. So a DNA polymerase in us, delta polymerase, repairs it, and it makes it double-stranded. So it uses that 3' hydroxyl at the end of the first T on the left to extend and make a fully double-stranded molecule. So now the cell is happy that this thing is double-stranded, but it has unwittingly made a template for transcription, right? So that molecule can be transcribed, and one of the proteins that produce is called rep7868, which is that orange oval there on the picture. That protein makes a nick in the double-stranded DNA where it says site of sequence-specific nick between A prime and D. That is nicked by this enzyme, which is made once the viral genome is transcribed into mRNA, and the mRNA goes out in the cytoplasm. It's translated to rep7868, then goes back into the nucleus, right? It nicks that, and then it remains attached to the DNA. It's like an origin binding protein, because that's the origin of DNA replication. And so now what happens is here's what the DNA looks like after it's been nicked. And then the T structure is denatured, and that's shown in the next panel here in step 4. So the blue part would be denatured, and then the polymerase fills in to make it double-stranded. So there really should be an extra figure here where you have just an extended blue, but that doesn't ever exist. In fact, what happens is the polymerase begins to copy at the 3' end, and it displaces the hairpin and makes the whole thing fully double-stranded. So now you have a complete double-stranded copy of the viral genome. And the ends can now form hairpins. They do that because the hairpins are more stable than the structure of the genome. And then you get reinitiation of DNA synthesis from that 3' end on the red strand. That's a 3' end. And you see the orange is now strand displacement of that bottom strand, the blue. We're displacing the top strand. So now we end up with a double-stranded molecule from that priming. And the top strand is displaced. It is a single-stranded molecule. And these can be replicated in the same way. And this double-stranded one can go back through the process and get a nick and go through this as well. So this one is also the polarity of genomic DNA, and that could be incorporated into particles. And so this is not semi-discontinuous. There's no replication fork. It's strand displacement, so replication is continuous. There's no need to make little DNAs because you can go 5' to 3' directly. There's no polymerase alpha. Polymerase alpha makes the RNA primers for SV40. You don't need that because the primer here is the inverted terminal repeat after you nick it. It does require other proteins in the cell, polymerase delta, to make long DNAs. The rep7868 is the viral protein that nicks the DNA and recruits the DNA synthesis apparatus. Okay, so this is strand displacement. And all these strands that are made will eventually be incorporated into viral particles. So it's an interesting way of getting around the end problem because you're priming right at the end. And there's no loss, and you don't have RNA, which you remove, so there's no end problem. Yes?
If you go back to the last slide, what's going on between 6 and 7? Where does the hairpin go?
Between 6 and 7, so the DNA synthesis is initiating at the 3' hydroxyl of the red, right? And that is what is shown in the next panel as orange. That's the new DNA that's being made, and it is displacing the top strand. All right, and that top strand ends up here, and the bottom one is double-stranded, and that can go through this whole sequence again. All right. Yes?
Can you explain again where the REP7868 coming from? Because you said it makes a transcription, but when it's elongating DNA, not RNA.
Right, so this, as soon as this molecule is double-stranded, it can be transcribed to make mRNA. And that mRNA encodes the rep78 protein.
So there's another process that happens
Yeah.
at the same time.
Remember, transcription always happens before DNA replication because you need at least one protein in order to get DNA replication. So the process we talked about last Wednesday, transcription always has to come first before DNA synthesis. Yeah. Okay? Adenovirus is a longer double-stranded DNA genome, has an origin of replication at both ends, which tells you right away that DNA synthesis is going to be beginning at both ends. And this is going to happen by strand displacement, as we will see. So we're going to displace the top strand, just like we saw with parvovirus. And there's also a protein linked to the 5' end of each strand. TP is terminal protein, and there's one at each 5' end. And that is the primer for DNA synthesis, a protein. We'll see that. All right, so what we start off with, the double-stranded DNA goes into the nucleus. It is transcribed to make all the proteins that are needed for DNA synthesis, all right? And then only when those proteins are made can DNA replication begin. So it's delayed because of that. But now we're at the point where we have polymerase. And so the replication, of course, is going to start at the 3' end of the genome. So here's a double-stranded DNA. Let's start at the 3' end here on the left, and we're expanding that on the left here. In this box, you see the polymerase, which is initiating synthesis, but it's utilizing a primer, the terminal protein linked to a C, a single C. It's actually in the form of pre-terminal protein. So what's on the DNA in the end that I showed you on the previous slide is the terminal protein. There's a precursor to that, which is a little bigger, and that's what's involved in the priming reaction. The pre-terminal protein binds the polymerase, and the pre-terminal protein has a C on it, and that acts as a primer. So the C base pairs with a G, that's 4 bases in from the end, and then the polymerase puts on A, and then T, and then it slips back 3 bases and continues. Why does it slip back 3 bases? Because it's hard to start at the end, at the very end of a DNA. So it's starting internally and slipping back. This is seen with a lot of polymerases. So now the polymerase can simply make that first strand, and it will displace the top strand. When that top strand is displaced, it's coated with a viral protein called DNA binding protein, the protein in yellow, to keep it single-stranded and not binding to any other strands in the cell. And eventually, the entire copying will be completed. So the copying of the, the copying, thank you, of the minus strand results in a red strand. And eventually you now have a double-stranded DNA, red and light blue. And this can go through that process over and over again. But you also have the displaced top strand, which is now coated with single-stranded DNA binding protein. But the ends of that molecule are complementary because they are inverted terminal repeats, just like the parvovirus DNA was. So they can form a double-stranded structure. And the polymerase recognizes that as if it were the end of a bona fide viral DNA. So the polymerase binds to it, it primes DNA synthesis, just as we see in these 4 boxes on the left, and it makes a complementary strand. So that will end up being a double strand, which can then go through that process again. So both strands are copied in separate reactions, essentially. And you have 2 DNAs where you had 1. This occurs over and over again. So strand displacement. Why is there no end problem? because we're not using an RNA primer that has to be removed and leaves a gap, right? We're using a protein primer that can slip back with the polymerase and prime exactly at the 3' end. And so there's no end problem because there's no gap that has to be fixed. This is an illustration of this single-stranded DNA binding protein. It's a viral protein whose job is simply to bind to the displaced strand. So what we have here is we have a template, parental DNA, double-stranded DNA. Here's a polymerase that's making double-stranded DNA from one strand. It's displacing the top strand, right? It's strand displacement. And as that top strand is displaced, it's bound up by this single-stranded DNA binding protein, which keeps it from base pairing with other DNAs that are in the cell. Obviously, there are a lot of DNAs, viral DNAs, floating around, including complementary ones. And so this keeps it denatured. And then that continues until that top strand is completely displaced and the ends form that double-stranded structure that I showed you in the previous slide. So the protein itself, the structure is shown here on the left in a space-filling diagram. You can see there's a core protein with a little extension of amino acids coming out, and we think that forms a kind of plow that helps denature the two strands of the DNA. All right, the third question is, how is DNA replication of parvovirus and adenovirus similar? They both require protein-linked primers. Replication occurs by strand displacement. DNA synthesis occurs in the cytoplasm. A replication fork occurs in both or none of the above. How are the two replication strategies similar? Okay, how did we do? Okay, replication occurs by strand displacement. That's what's similar between the two. They don't both require protein-linked primers. One is a DNA primer, one is a protein primer. So the parvos are DNA, that little hairpin. So a lot of you pick that. So I'm going to go back to the slides and show you. And then DNA synthesis occurs in the nucleus for these viruses. And a replication fork occurs in neither, right? They're both strand displacement and none of the above. So the protein-linked, which one was I going to show you? The replication fork occurs in both. All right, let's look at both of them. So here's adenovirus. We have strand displacement. There's no replication fork. And it's protein-linked. And for parvovirus, again, we have strand displacement. And the primer is this 3' end. The primer is the 3' end of the hairpin, which is made by a nick by the viral endonuclease. So a DNA primer versus a protein primer. But they're both strand displacement. Moving to bigger molecules, herpes simplex virus. This is a rather large DNA virus with a double-stranded DNA of, what is the size? 135,000 base pairs? You know what? I don't remember. And neither do you have to remember, but I want to know. I would never ask you how long a genome is. Yeah, 150,000 base pairs. Double-stranded DNA, linear. It has three origins of replication, actually, ORI-L, ORI-S, and ORI-S. And the DNA will come in the cell as a linear molecule, goes into the nucleus, it gets circularized, as you'll see in a moment. So we may say, why does it have three origins? Probably to accommodate different ways of replication. So this virus has a latent period where it's not replicating, and then it has a period where it is. And so it happens in different cell types. So probably those different origins accommodate all of that. Now, this is one of the viruses that encodes all the machinery to make its own DNA. And you can see these proteins up in the upper right. There's a primase to make RNA primers. there's what's called a processivity protein, which just helps you to make long DNAs. There's an origin binding protein. There's a single-stranded DNA binding protein to keep the strands separate. And of course, there's the DNA polymerase. So this happens by the DNA coming into the cell as a linear molecule. If you remember, the virus fuses at the plasma membrane, and then the nucleocapsid comes out and moves to the nucleus, docks on the nuclear pore. The DNA comes out of the portal into the nucleus. In the nucleus, it's circularized by a host protein called DNA ligase 4, which takes the two ends and makes a covalent bond. So now we have a circular DNA in the nucleus. And that circular DNA reproduces by rolling circle replication. So to do that, you have to make a nick, which is carried out by an endonuclease. It's a cellular endonuclease. So now you have a 3' hydroxyl that you can start to add bases to. You always need a 3' end. And you don't need an RNA primer because, at this point, because you're using DNA as the primer. So the DNA can be made by DNA polymerase. It's red here, and it starts to go around the circle. It will displace the top strand, and it keeps going around and around. Now the top strand that has been displaced is shown in this figure as coming off to the right. And you can imagine this circle rolling as the DNA synthesis occurs and more of that original strand comes out. That's called, so that's part is called continuous DNA synthesis because the polymerase starts at the 3' end and just keeps going. It never has to stop. Whereas on the other strand, the polarity of the template is wrong and it cannot do continuous synthesis because you can imagine if only a little bit of this strand were exposed, you could start synthesizing there with an RNA primer. And then when more of the strand comes out, you make another one and another one. So that's discontinuous. It's like our SV40 discontinuous strand. And that keeps going, and you make what are called concatamers, which simply means long DNAs with many genome lengths in them. Genome length meaning the length that can be, well, 150,000 base pairs. And you make multiple genome length molecules. These are repaired, the RNAs are removed, and the DNA is filled in and ligated, so there's no end problem because you can continuously fill in those gaps. And these are made in very long molecules, and it's not until they're packaged into new capsids do they get cleaved. So they exist in the nucleus as really long DNAs encoding multiple genomes in them. All right, so that's what rolling circle is. One year, one of the students said, "Isn't that like toilet paper?" Mm-hmm. Yeah, it is, kind of. Each sheet being a genome. It's not double-stranded, of course, but you get the idea that the toilet paper unwinds and you get these multiple genomes coming off, right? So you can think of it that way. Toilet paper replication. All right, that's for herpes virus. And finally, our last virus, pox viruses, which are different. They're big DNA genomes, over 250,000 base pairs in length, double-stranded DNA, which are really single strands, but they're base paired with each other because the ends are covalently linked. If you melted this DNA or denatured it, separated the two strands, it would be a single-stranded circle, but it exists in the virus as a double-stranded DNA with the ends linked. And they're inverted terminal repeats at the end. Now, this one we're talking about because it replicates in the cytoplasm. It's the only one so far that we've talked about that replicates in the cytoplasm. All the others go in the nucleus where they can access other proteins from the cell that are needed for DNA synthesis. But this one is in the cytoplasm, so it has to make all the proteins needed for DNA replication. It also has to make everything you need for transcription, right? The DNA-dependent RNA polymerase to make mRNAs, because you need some proteins before you can make DNA. And this virus needs a lot of proteins before it can make DNA. It's got to make the whole machinery on its own. So that's quite a challenge. But apparently it works. it's evolutionarily competitive because there are lots of viruses like this. Many of the giant viruses also reproduce in the cytoplasm. So here's an experiment showing cytoplasmic replication. So we have cells infected with a pox virus, and we're staining them with DAPI here on the left. That's a stain for DNA. So any DNA will be blue. And you can see there's the nucleus, obviously, but the cytoplasm also has some DNAs in it. And this is a virus-infected cell, so that's actually viral DNA in the cytoplasm. And how do we know that? Well, then we stain separately for DNA binding protein. And remember, that's a protein that's going to bind to the single-stranded DNAs to keep them from reannealing. And that looks like it's outside the nucleus, which you can confirm by merging the colors from A and B here in the merge. And you see the DAPI nucleus is here. These areas are outside the nucleus, but they co-localize with the DNA outside the nucleus, which means those are what we call factories where viral DNA synthesis is occurring. So you see there's no signal for DNA binding in the nucleus. That tells you there's no nuclear DNA replication. It's all in the cytoplasm. Now the way this DNA replicates is unusual because it's a covalently closed circle. So what we think occurs is, so we have our double-stranded genome at the top there. That's what's in the virus particle. And then we believe at one end, the replication initiates by the synthesis of RNA primers, right? Because there's no primer for synthesis in this molecule. We're not cutting it or anything. We synthesize 2 RNA primers, and then from them, we prime DNA synthesis. So now eventually, that DNA can go all the way around the circle in one process. It's continuous replication. And now you have a dimer. And somehow those 2 strands have to separate because the viral DNA in the particle is single-stranded DNA. So how that happens, we don't know. But it's called concatemer resolution, very much like what we saw for SV40. The topoisomerase cuts it. We don't know if that's what happens here. And you end up with 2 monomers which go into virus particles. And there's no end problem because it's a circular genome. So you don't have that issue. These 2 priming initiating steps here, when the DNA comes back to them, the RNA can be removed and the gaps can be easily filled in without losing any sequence. And our last question is, what makes poxvirus DNA replication different from all the other viruses we discussed today? The DNA polymerase requires RNA primers. DNA synthesis occurs in the cytoplasm. DNA synthesis occurs by strand displacement, none of the above. Okay, let's see what we did. DNA synthesis occurs in the cytoplasm. That's what makes poxvirus DNA replication different from all the others. Other DNA polymerases require DNA primers, RNA primers. Not all of them, but others do, so that's not different. Strand displacement, nobody picked, none of the above. No, the cytoplasm is what makes it different. Let's talk a little bit about viral origins, not where they came from, but the origins of replication. Origins of replication, we've talked about for SV40, we've talked about for adenovirus, the ends are the origins. For herpes, they are the ORIs, the 3 origins here. For parvoviruses, the ends where the Rep6878 protein binds and the polymerase binds. These are typically AT-rich segments recognized by binding proteins. They're AT-rich so that they can be easily denatured. And they are where the DNA replication machine is assembled. You can have one, and you can have more origin, as we saw for herpes viruses. Here are some comparisons of 3, SV40, herpes, and adenovirus. So we have, in SV40, we have large T binding sites, as you can see. Those are the yellow colors, sequences bound by origin recognition proteins. We have SV40, we have for one of the herpes origins, and also the end origin, one of the end origins for adenovirus is also bound by the origin recognition protein. Then we have AT-rich sequences, which makes it easy to denature. These are the kind of greenish sequences you can see in all the origins. And interestingly, these origins of replication typically overlap with transcriptional regulation sites. So they're often promoters located in these regions. So for the SV40, the early and late promoters go in both directions from the origin. For herpes, you can see there are promoters going in different directions around the origin, and the same for adeno, but it's not shown there. So for some reason, they overlap the origin sequences and those for transcriptional regulators. Maybe it's to keep the information compact. These origins are recognized by viral origin recognition proteins. We talked about T antigen, which binds to the origin. The parvo rep68 binds at the ends and it nicks and helps unwind the DNA. The adeno pre-terminal protein is the origin binding protein that binds at the ends, and it recruits the DNA polymerase. And there's a herpes protein called UL9, which also binds to the ORI as well. So in each case, you have an initiation site for DNA replication and a viral protein that binds it to get the initiation, to get the replication process going. Here's a diagram of SV40 large T, right? The origin binding protein for SV40, which has been described as the most studied protein in the world. Every amino acid of this protein has been changed in multiple laboratories. It's a 708 amino acid long protein, and that bar is the protein there in brown. And you can see it has all kinds of domains in it. Let's see, for example, the part of the protein that binds DNA there, origin DNA binding. There's a nuclear localization signal because this is made in the cytoplasm, has to get in the nucleus. There are sequences for binding the polymerase alpha sequence binding regions there, and also at the N-terminus. Part of it is an ATPase because you need ATP hydrolysis to unwind DNA. It's a helicase. It denatures DNA, and there's the activity for that. So you can see lots of different parts of the protein have been identified by changing the amino acids. This protein also binds to cell cycle regulators to push the cells into S phase, and I'll explain that in a moment. This T antigen is also a protein that controls what species the virus will reproduce in. So SV40 stands for simian virus 40. It's a primate virus, will not reproduce, say, in mouse cells. And that's because the T antigen does not interact with DNA Pol alpha primase in mouse cells. It only does so in primate cells. So it explains the tropism of the virus, the species that it's able to infect. But let's take a look at the cell cycle. What do I mean by it pushes the cells into the cell cycle? Well, most of the cells in your body are not replicating, but there are a few examples. Most of your epithelia are reproducing in respiratory tract, alimentary tract, urogenital tract epithelia. They're dividing and come off periodically. Your skin is always dividing, and the white stuff you see floating around in rooms, that's your skin mostly floating around. When you dust furniture, what you're dusting is your skin mostly that falls off. It's a nice thing to think about, right? But most of our other cells aren't replicating. Your liver, your muscles, unless you're working out, your brain cells, they're not replicating. But DNA viruses cannot reproduce in cells that are not replicating. Most DNA viruses cannot. Because, why not? Because they need DNA synthesis proteins from the host, and unless the cell is dividing, those proteins are not available. So viruses, when they get into a cell that is not replicating, they have to make it replicate. They have to induce host replication proteins by making the cells replicate. So if a virus gets in a cell which is not dividing, the virus makes proteins that will cause the cell to divide, and that means it's going to turn on DNA synthesis proteins. And it is the early gene products that does this. So the SV40 T antigen, right? That one protein that's made early, that makes cells divide. The adenovirus E1A protein, the papillomavirus E6, E7 proteins. They are the ones made early, and they make the cells divide. And they do so, again, because you need to make DNA replication proteins to replicate the viral genome. This has huge consequences when we start later to talk about viruses causing cancers. This is going to be what's going on. But for now, let's look at how this works. Here's the cell cycle, right? It's highly regulated, roughly 24-hour cycle in most cells. You have a period of mitosis where the cells divide, and then you have two daughter cells. Again, they're called daughter cells. Then those cells are pretty small. They're half the size of the original cell, so they grow. They have to get bigger. That's the G1 phase. Then they replicate their DNA. That's the S phase. And then they get ready to cell division, and they divide again in M. And division is, of course, called mitosis. That's shown at the top. But this cell cycle is highly regulated. There are multiple checkpoints in a cell to regulate it. And one of them is called the RB protein. So here is a restriction point in the cell cycle, which is controlled by the RB protein. RB is a gene discovered in retinal tumors of young children. They are found to have deletions or loss of this gene, so they develop these tumors early in life. And it turns out there's a protein encoded in that gene that is a checkpoint for the cell cycle. In other words, if conditions aren't right for cell division, if there aren't enough nutrients, the RB protein will halt cell division. So most cells do not divide because RB protein is holding them in G1. And so the T antigen of SV40, for example, the E1A of adeno, they abrogate RB protein to make the cells divide. And that's shown here. So the way RB keeps cells from dividing is shown here. You need a lot of different proteins made for cell division. And those are encoded by multiple genes. And those genes, of course, are transcribed by a promoter. So here on the bottom right, genes encoding cellular and some viral replication proteins. These are needed for DNA synthesis and to pass through the cell cycle. So multiple genes. And RB prevents their transcription and thereby halts cell division. These promoters require a cell transcription protein called E2F, which normally would bind these promoters and activate them. But when RB is bound, it blocks the transcription of these genes. And so RB being bound to E2F prevents genes being expressed that are needed for cell division. Okay, so E2F proteins are needed for transcription of genes needed for cell division and DNA synthesis, and RB blocks that. Yes, so the question is, could this make non-permissive cells permissive? Yes, if a virus cannot replicate in the cell because the DNA machinery is not available, that's a reason for not being permissive. So this would change that. It would make them permissive, yeah, because it now turns on synthesis of proteins that are needed for DNA replication.
That's not the only case that non-permissive cells.
No, there are a lot of other reasons too, right?
Okay.
This is one example, yeah. Okay, so when SV40 infects a cell, or adenovirus or human papillomaviruses, the early protein is T antigen or E1A or E6, E7. That binds RB. So this little protein up here, the pink one, is large T or E7 or add E1A. This binds RB and prevents RB from inhibiting the promoter transcription. And so now you have E2F binding these promoter regions, and they can be transcribed. They're no longer repressed by RB protein. So the cells divide, and they make DNA synthesis proteins which allow the viruses to replicate in them. And eventually these viruses kill the cell, so there's no issue there. But sometimes the cell doesn't die, and these T antigen genes become permanently expressed, so the cells divide forever. They become immortalized. And HeLa cells, which came from Henrietta Lacks in 1951, right? She had a tumor of her cervix. They took it out, and put it in culture, it grows forever. Those HeLa cells are growing today. The reason is she had a human papillomavirus infection, and the HPV E6, E7 gene became integrated into her DNA and made E6 continuously, and those cells in her cervix divided forever. And when you have division forever, that's how you get a tumor. That's how you get cancer. We'll talk about that later. But that's why cells do not divide forever in us. Because if they do, they start, in every cell cycle, a cell makes mutations because DNA polymerases make errors and they accumulate, but eventually the cell dies. But if cells divide forever, those mutations will accumulate continuously, and when you get mutations in about 12 different genes, then you have a cancer. And so viral proteins like these can do that. And this gave us, and we're going to talk about this separately, but this gave us a lot of insight into how cells become tumorigenic. All right, so next time we're going to talk about this very interesting family of viruses where their RNA is the genome and it gets converted to DNA by a process called reverse transcription.